So in today's class, we'll be discussing about DNA motif discovery using the popular greedy algorithm. So as an introduction, these are some prior knowledge which we may need to understand the greedy algorithm for motif discovery much in a better way. And for that, you have to understand what a motif is. Motifs are really short recurring patterns in DNA which is biologically important. Okay, so what does this mean? So motifs are patterns that occur frequently in a DNA. It may be in a single DNA, or it may be that we are comparing DNA of two organisms and we are looking which all patterns come in common between them. But there is an issue in biology and especially in genome. The issue that we are going to face will be the patterns will not exactly come as similar patterns, because DNA has a process known as mutations and there are high chances that one or two nucleotides may actually change. But the overall pattern may look really same, but there may be a small fraction of the nucleotide which may undergo a change. So motifs are they are short. They can be of lengths around like 10 or like 14 nucleotides. They are recurring. They not only occur once, but they keep on occurring here and there, and they are biologically important. But in the computational approach, we will be only considering motifs as short, recurring, similar patterns. Then what is a greedy search? Greedy is, as the name suggests, is an important algorithm in which greedy has some qualities, like it does not go and search for all possibilities of a particular thing we are searching for. It only searches for like it in the process of searching, greedy will only consider some initial conditions and it will always pursue in that direction. Of course, it may or may not lead us to the global solution, but of course we may get a little bit what important local solution which can resemble the global solution, especially when it comes to DNA. But it doesn't matter whether it's a local solution or a global solution, both is going to be similar. So in greedy search, what a greedy search algorithm, what it basically does is that it chooses the best in each stage. There is no backtracking. It is a local solution. It is not an exhaustive search. By exhaustive search, I mean it does not go and search for all possibilities. It will just consider one or two initial possibilities and will come at a conclusion. Why are we using greedy search? Why can't we use some search which uses exhaustive means, like it goes and search for each and every possibilities? Actually, this is why we are using greedy search. The greedy search has very less time to give the results. Remaining algorithm, mainly the deterministic ones means it goes on search for all possibilities. It doesn't employ any probability concept. We will be getting the global solution in that. But the time will be really long. It may take hours or days to give the result because genomes are really long. So that's why we are using motif discovery using greedy. It takes very less time and also results closely resemble the node motifs. We will be getting a pattern which we will be getting a set of patterns that are extremely similar, and that's more than enough because we are only looking for motifs which we do not know how they look in prior. We just run this algorithm and get a set of patterns that look extremely similar. Okay, this is a common methodology. Maybe you will not be able to understand this in the first view, but once we come into the implementation level, you will understand how greedy is employed to search for the motif. Well, there will be only two inputs and only one output. The two inputs include the DNA sequences, which DNA sequence we are considering, and then also a k length will be given. This k length is actually the motif length. Maybe motif length can be 12, motif length can be 10, whatever, according to the organism or according to the needs, we may design the k length. DNA sequence is given as an input. So the first step will be, we will be forming the DNA matrix. What the DNA matrix means is that what a DNA matrix means is that if I give you a set of DNA or a single string of DNA, then to make the process easier, what we will be doing is that we will be converting that into a matrix. Matrix in the sense we will be dividing the DNA at equal intervals and store the respective parts as in a matrix form. This will be part number 1, this will be part number 2, and there is a condition to this: all DNA strings must be of equal width. So maybe some extra will come here that we may just ignore. So this is what you know as a DNA matrix. Actually this DNA matrix is really needed for the further computations. So that comes in the forming the DNA matrix stage. In the next stage, we will be selecting the first scene k-mers to create the profile matrix. Actually, this is the reason why this algorithm is known as the greedy algorithm. It will only select the first scene k-mers to create the profile matrix. So what is a profile matrix? Profile matrix is just an evaluation criterion. So it is a means of evaluation. What is that? We will come to it. So it is a means of evaluating k-mers. So it is a means of evaluating the k-mers. What does this mean? Evaluating the k-mers means if I give you two k-mers, you must say which k-mer are you looking for, 1 or 2. So to evaluate that process, we will be using the profile matrix. How will we create a profile matrix? We will come to that. So selecting first scene k-mers is actually the greedy approach. By greedy approach, I told you we will be arriving the conclusion by seeing the first few things. That is why it is known as greedy. It does not have enough patience to go and look for the remaining possibilities. So in the second step, what we'll be doing is, I told you there will be a k length. Let's take it as, for example, let it be a 9 k-mer. That means that particular pattern will be a continuous pattern of 9 nucleotides. ACGT, like that will become 9 nucleotides. So selecting the first scene k-mers means I have the first string of DNA here, which can be of length maybe 200, 300, etc. So I'll be taking the first 9 from first till 9. Okay, I'll be selecting this k-mer, and similarly from the second row also the 9 nucleotides I'll be selecting. From the third row also 9 nucleotides I'm selecting, and these many I'll be storing into a matrix like thing. Okay, the first 9 mark, second 9 mark, third 9 mark, similarly so on. Okay, so this is actually a new matrix, right? But this we don't want to form it as a matrix, but we have to convert it into a profile thing, a means of evaluating other k-mers. So how is this done? Let's see. So how do we create a profile matrix is the next question. So what we have is a DNA matrix. I'm telling in general. So this is a general way of telling how you will create a profile matrix or an evaluation criterion. So I am having a DNA matrix. Okay. So this is DNA matrix. Now the next step will be I'll be forming a matrix known as count matrix. What a count matrix will contain, it has a peculiar quality that a count matrix will only have 4 rows. In case of DNA count matrix, okay, only 4 rows, 1, 2, 3 and 4. So a count matrix will only have 4 rows, and the first row will represent the nucleotide A, the second row C, the third row G and the fourth row T. So A, C, G, T, these are the rows in the count matrix. So what we will be how we will be filling the count matrix is that we'll be looking the DNA matrix. So DNA matrix may appear like this: A, C, G, etc. And the second may be T, C, A, etc. So it goes on like that: G, A, something, some arbitrary DNA sequences. It goes on like that. So in count matrix, we'll be taking the first column of the DNA matrix and count in that column how many A's are there. Currently, A only 1 is there. How many T's are there? Only T1 is there. How many G's are there? G only 1. Is there any C here? No. So C is 0. If there were 2 A's, what will be done? 2 A's. So there is a peculiar property of the count matrix that the number of rows will be the number of unique nucleotides and the number of columns will be same as that of the DNA matrix. DNA column numbers. How many DNA columns will be there? That many count matrix will also be having those many columns. So this is how you form a count matrix. So we arrived at a stage. Now we have the DNA matrix. From the DNA matrix, we formed the count matrix, right? By just taking the frequencies of A, C, G, and T, and taking the respective counts from each column and updating here. Like that, we'll be getting the count matrix. And once count matrix is there, profile matrix is really simple. Actually, there is no much difference between the profile matrix and the count matrix, but profile can be called as a normalized version of the count matrix. Normalized means it's like this: one element divided by the total element. That's what you call as the normalizing process. And profile matrix is simply the normalized version of the count matrix. That means the dimensions of profile matrix and count matrix will be exactly same. If this is n by m, this will also be n by m. This is ACGT. Similarly, this is also representing ACGT. Actually, I'm just writing this. This does not form as an entry. This is just like a heading we are giving. And then there will be values here, right? First column there may be like, okay, maybe 0G, maybe 0C, maybe 8G, 3T. So what I'll be doing is, like that there will be elements here, for example 4, 3, etc. So in the count matrix A, what I'll be doing is, I'll be writing 1 divided by total. What is total? 1 plus 0 plus 8 plus 3. 8, 9, 10, 11, 12. 1 by 12. See, like that I'll be filling. The next will be 0 divided by 12. The next will be 8 divided by 12. 3 divided by 12. It goes on like that. So this basically means, on average, how many A's can you see, or what is the probability of finding A in the first position of the DNA matrix, right? Then similarly, next value will come here, maybe 3 by 12. So we can tell that the probability of finding A in the second location is 3 by 12. So we will be getting a measure to tell what is the probability to find a particular thing in each column. So like that we will be getting the profile matrix. So this simply is a profile matrix. So this is why I called a evaluation criterion. Okay, so if I am giving you this profile matrix and I am asking you what is the probability of ACG and GAC, actually you will be getting different values. How you can evaluate? Okay, maybe this is only a, what is it, maybe a 4 by 3 matrix. I am going to fill values 4 by 12, 3 by 12, 2 by 12, 5 by 12, etc. Okay, so how I am going to find the evaluation for ACG and GAC based on this profile matrix? So first ACG. So first position is A, and okay, so the value is 1 by 12, multiplied it with the second position is C, but this is A's value, I want C's value. C's value is here, so it is 3 by 12 into the next is G's value here, maybe it is 5, again 5 by 12, so it is into 5 by 12, sorry, it is 5. Okay. So you'll be getting a value. Similarly for GAC also we can, so G's first position is 8 by 12 into the A's position is 3 by 12 into the C's position is 2 by 12. We'll be getting a value here. Clearly we can see the value is greater for GSC, therefore GSC qualifies as a candidate from the evaluation and ACG does not. 5 into 3, 15, 24, 24 into 2, 48. So this is why I called profile as an evaluation criterion. So from DNA, if we create count, then profile is really easy to compute. It is just a normalized version of the count. Okay. Keeping all these things in mind, let's move on to the algorithm. So we form the DNA matrix. We select the first k-mers from each row to create a profile matrix. So what does this mean? We have the global DNA or the main DNA matrix here, and then we selected the first k-mers, created a new DNA matrix. Okay, these are the small k-mers. From this we form the count matrix, not from this. From this we form the count matrix. From that we created the profile matrix. Okay, now we have evaluation profile. Evaluation criterion is there, that will be the profile matrix. Next what we'll be doing is selecting the k-mer motif 1 with highest score from first row. Okay, what does that mean? So now this is no longer useful. This and this is no longer useful. We only want a profile. We use this profile to evaluate each k-mer in the first row of DNA, the main DNA matrix. So k-mers will be selecting like this. So if it is A, C, G, T, A, C, G, I am asking you to select all k-mers of length 3. So it is ACG, CG, CGT, GTA, TAC till the last. Okay. So we'll be extracting each k-mer and send it to evaluation using the profile matrix. We'll be getting a value. Similarly, the next k-mer will be sending it to evaluation profile. Will be getting another value. Like that, we will be doing for all. Okay. And the one with the highest score or the highest probability, for example, let it be TAC, that will be selected as our first motif, fixed. Now that will never get changed. That means we fixed this as a motif 1. Okay. The first motif is this, we have fixed it. Now what we do is that we are going to change our profile matrix. No longer the evaluation that we used using the greedy method. Why I call this greedy method? Because without considering anything, we just took the first k-mers and created a profile. Therefore, that is a greedy method, right? We did not look for any other possibilities to select the best k-mer from the first row, and that which we named as motif 1. Now no longer we will be using this profile because this profile is not a good measure. Instead of using this profile, we will be creating a matrix known as motif matrix, into which the first motif will get added. So now the motif matrix will only have one entry, which is TAC. Now from this we will be creating the count matrix. Okay, it is easy to create a count matrix. If it is A, C, G, T, in the first position A is 0, C is 0, G is 0, T is 1. Similarly 1, 0, 0, 0. Again 0, 1, 0, 0. See, this is the count matrix. From this we create a profile matrix. Now when you will doubt, because 0 divided by 1 will go out 0, 0 divided by 1 will again go out 0, 1 divided by 0, everything will go, this will go infinity and all. So many issues because of occurrences of zeros, right? Anyway, let's forget that issue for now and let's assume that we get a value. Assume that we'll be getting a value for each of these entries. We'll be handling this 0 case later. I'll be talking about that, don't worry. So for currently, you only remember that we create the first motif place it in the motif matrix, create a count matrix, and create a profile matrix. Okay. Now we have a new profile matrix. What we will do with this profile matrix? This profile matrix will be used to evaluate all k-mers in the second row of the DNA. Okay, just like we did for the first row with the greedy profile matrix. Now we are going to evaluate the second row with a evaluating this motif matrix. Okay, thus we will get a motif from that. Okay, once we get a motif from that, we will be adding that to the motif matrix ACG. We will not delete the first one, we will just add to it. Now using these two, we will be creating a new count matrix, a new profile count matrix, a new profile matrix. Okay, and then using that updated profile matrix, we will be evaluating the best schema from the third row. And then we will get again one more new motif from there. That new motif will be adding to the motif matrix. This process will keep on adding. So how many rows this DNA matrix have, those many entries in the what this motif matrix will also be added. That means that many motifs we will get. So if we want to get 12 motifs, then that means then your DNA matrix must have 12 rows. Okay, this is the overall working of the greedy motif search. So you form the DNA matrix, you select the first scene k-mers to create a profile matrix. This step is the greedy method. In the remaining part, you will not use this step. This step is only for once you will be using this step. Then you will select the k-mer motif 1 with highest score from the first row using this profile matrix. Then you update the motif matrix with the first motif. Then you keep on repeating this for every other row. Each time you will get a new motif, you will add it to the motif matrix, create a new profile from that and evaluate. That's how you keep on running till the last row of the DNA matrix is reached. So, and at last you will be returning the motif matrix, which will contain different motifs, motif 1, motif 2, motif 3, each extracted from each row of the DNA matrix. So the output will be a set of similar patterns. So this is a methodology in the greedy search algorithm that we employ for motif finding. So I have just to show how well this algorithm works. Well, this I implemented the greedy algorithm and ran it through the search ran it through the genome of Vibrio cholerae, the pathogen that causes cholera, and I got these motifs. Don't they look exactly similar? Yes. See the patterns. See how T is repeating. See only one. I told this is a complexity in motif finding. See one nucleotide change, but remaining all it's extremely identical, right? Not one. Okay, two mutations had occurred. See like that. Isn't this all these similar? Yes. So this is an output from the greedy algorithm. This is how it looks. See a matrix. This is a motif matrix, a final motif matrix. So talking about the concluding remarks. So greedy search is an effective, very effective. You saw the results. It is cost effective in terms of time. Cost effective means time it takes really less to compute. Maybe some 1 or 2 seconds, not more than that. So it helps to find very similar set of patterns which can be eligible candidates for motifs in DNA. Whether it be a motif or not a motif, then it's up to the biologists. They need to conduct experiments to see whether they are biologically significant. But as a computer scientist, we just want to find the similar patterns. The search can be performed for any desired length from big DNA sequences. So even if we give 1,000 million DNA sequence, it can be able to compute with very less time. So that's all about greedy algorithm. Now I'll be showing you a demo in Python 3. See, that's only applicable if you are known, if you know some basics in Python, you want to watch that. Okay, I will not be taking very complex things. It will be very simple logic or simple coding style that will be employed to show you how the greedy algorithm can be implemented and ran to give a successful result. I'll be showing that, okay, in the next part of the video. So this is where I am going to show you how we can implement the code in Python. So this is just a Jupyter notebook platform in where I am going to write blocks of code and run it for better understanding. Okay, so first, as I told, I'll be creating a DNA matrix. How will I create a DNA matrix? I just need two entries. One is DNA and a row size. So DNA matrix being an empty list, I'm running a loop through. So the basic logic is this: I'm going to cut the DNA at the desired row size. If I tell you 10, for every 10 nucleotide reach, I'll be cutting there and giving it as a first row, second row, appending all rows. Simple code. If you go through it, you will understand. And thus at last I'll be returning the DNA matrix. So here I have ran it as just a small example. So this is a DNA matrix. I gave the value 3 and that it got split at 3 positions. See ACG, CCT, like that got split. Now this is the count matrix. Okay. In the count matrix can only be created if you give in a DNA matrix. So giving in a DNA matrix, okay, this line is just I'm taking the transpose of that matrix for easy computation. Okay, now there is something known as Laplace's rule. I told you that that's zero issue, right? Because that zero come, we cannot divide and proceed. Everything will get zero. So we need a condition that the minimum entry in our DNA matrix should be 1. 0 should not be an entry. So we will be initializing all the counters with 1. It doesn't matter because the priority will go if you see a repeated nucleotide, one will get incremented. So whether it's 1 or 0, it doesn't matter. That's what Laplace's rule means. So you initialize the counters with 1 and then you search. If the first one is A, then you increment the counter A, then otherwise counter C, otherwise G and counter T. Alias you are appending all to a list and you're resetting counters back to 1 to go for the next iteration. And then at last you are taking back the transpose because you initially took the transpose and you're returning the count matrix. For example, if this is how you denote matrices. Okay, in Python, this is better to denote matrices: the first row, second row, third row. So you have to visualize like this: ACGT as the first row, second row is AGCT, like that. And then when you count, A is happening in the first place 1, 2, only 2, but it's written 3 because I have applied Laplace's. 3 plus 1, sorry, 2 plus 1, 3. Similarly, each row this will represent A, this for C, this for G, and this for D, as I told there in the lecture part. Similarly, a profile matrix is just you take the sum total of the first row, 3 plus 2 plus 1 plus 1, divide all elements with that. It's a simple program here. Well, I think I gone in an indirect way, but you could implement this in far better way. I implemented this so that you could understand it better. Just go through this, you will understand. See, I am converting each to integer because all these are strings, and then divide by the total. Thus profile matrix I'll be getting like this. Actually this much is the first row, actually it doesn't have space, so that's why it's printing like this. So this one is the first row, this one is the second row, like that will go. Okay. Next I have a function kmer profile value. That means if you give me a kmer, that means a short pattern maybe of length 10 and a profile matrix, I have to return a value for that. I told, right, multiplying that 1 by 4 into 3 by 5 into that's how I'm calculating. So multiply, initializing with 1, counter and all is there. So okay, a global nucleotide, this will actually store the value. Okay, that 5 by 12 into 2 by that, it will store. So it takes the value from the profile. It takes the value. Profile 0 means A, profile 1 means G, C and T. Actually count will maintain the jumping from one column to the next, and 0, 1, 2, 3 will maintain if it is A, C, G or T. After each iteration we increment in the counter so that we jump to the next row. And we'll be multiplying the final result and returning that multiplied value for that kmer. Okay. For example, I give this value ACT and I ask this is a matrix, you form the DNA matrix, you form the count matrix, you profile matrix. So it's basically I want a profile matrix. And I gave a kmer. What is the value of that kmer? The value came out to be this. Selecting first k-mer. Actually, this is the greedy part. We should select the first k-mer. Only the first k-mer from each row of the DNA matrix. So it's a simple algorithm. For example, this is a DNA matrix and the first k-mers are ACG, AGC and GCT. That's what I got here. All k-mers. So if I give in a string of DNA and k length, which are the all possible k-mers. So there is a rule like this. It will be always from 0 to length of the string minus k plus 1. This is a formula. The possibility of the last k-mer will be at this location. After this, if I take, it will break, like only AC will come. See, because that is the last one. See, so I want to stop things here so that I'll get a compact 3 k-mer at last. So this is it. So first ACG will come, then CGT will come, like that. In short subsequence things will come up. Yeah, okay, why put DNA length minus k plus 1 to get the last k-mer and not go and get AC, C and all. To avoid that, to stop there, I have used that formula. And then the best probability k-mer. So if I give you all k-mers and a profile matrix, what you have to do? You have to evaluate each k-mer and take which has the best value. That's why I created this method there, that k-mer profile value. I'll be evaluating for each k-mer. For I in all k-mers, I'll be evaluating profile matrix. Store the value to dictionary and get a value. If greater than the good value, then I'll be replacing that and returning that value from the dictionary. For example, this. Which one has the highest evaluation criterion? So that this list I am passing as input. So I got that CCT as the highest evaluation criterion. Now this is the main greedy algorithm. Okay. So initialing a list motifs. I'll be creating the main matrix, which is a DNA matrix. Selecting the first k-mer. That this much is the greedy approach. Okay. Selecting the first k-mers from that matrix, and for that k-mers I'm creating a profile. This much only is the greedy method. Remaining all are just the computational base. Then the first row all k-mers from the first row I am taking, forming the okay, I'm taking the first all k-mers and taking the best probability k-mer from the first row and giving to motif 1, and to the motif list I am appending motif 1. Similarly, counter 1 I am keeping just to maintain track that I go through all rows of the DNA matrix. 0 to length of matrix. Length of matrix will give you basically the number of rows in the DNA matrix. You'll be creating a profile. Now onwards from motifs. First you may create from the first k-mers, but now onwards you'll be creating from motifs for the remaining iterations. The best motif, each time you get a new motif, you'll be appending it to the motif, and then now the motif will contain 2 elements. That motif will be using here. Like we'll keep on repeating and returning the motif. So you can avoid this. Okay, now here, okay, actually the greedy algorithm should take 3 inputs. K is the motif length, DNA is the DNA full DNA string. The row size is the DNA matrix row size. If you want in each row of the DNA matrix 12 elements, and you could define here 12. So this is greedy algorithm. This is actually the entire genome of Vibrio cholerae. So I have to give 13,000. That means each row will have 13,000 nucleotides. Otherwise this list will be too long. Okay. So and 20, that means the length of the k-mer is 20. So I extracted these are the final motifs. So there are 1 2 3 4 5 6 7 8 motifs, because if I want more motifs I just want to decrease this value, so I'll get more motifs. What's happened when I decrease this? Yeah, more rows will get fit into DNA matrix. More rows in DNA matrix means those many rows will be there in motif matrix also. See, they are extremely similar, right? So this is how you get working for, actually this is not Vibrio cholerae, this is some other organism. I just kept it to show you. So you'll get similar pattern. So this is how greedy algorithm works. So hope you understood. So if you have any doubts or issues, please remember to post them in your comment, and if you really like the video, do like, share. Yeah, that'll be great support to all of us. Okay, if you have any confusion, do report it in the comment section. I'll really try to answer it whenever I get time. Okay, thanks for watching.